Hello, and thank you for joining us today. Welcome to our webinar entitled Development of HTS Enzymatic Assays for RNA Helicases, DDX3, DDX5, DDX17, RIG1, and MDA5. I'm Justin Brink here at Bellbrook Labs. Please note that if you have any questions during the webinar, you can type them into the chat box. We'll do our best to answer them during the live discussion. I'd like to introduce to you our presenter, Mahabat Ali, Senior Scientist at Bellbrook. He has a PhD in biochemistry from the University of Wisconsin-Milwaukee, where he studied enzymology. Mahabat was instrumental in developing these assays, and I'm excited to hear about his work, which he will share with us today. Thank you. And with that, here's Mahabat.
Hi, thank you, Justin, for the introduction. I'm super excited today about this webinar. We will be sharing about the development of high-throughput screening of enzymatic assay for some RNA helicases. So for the target-based small molecule inhibitors, it is very important to get a sensitive and robust assay platform for the assay screening to get the hit compounds. Recently, DDX protein or RNA helicase enzyme is getting interest and attention from the scientific community for its huge potential for the drug development target. Here we have development, as we see, the assay, high-throughput enzymatic assay for several DDX helicase enzyme using our Transcreener assay platform. So, before going to our discussion, here I would like to show you what would be our point of discussion today. The presentation will be around 20 minutes, more or less 20 minutes discussion, and after that I will be happy to answer all of your questions. So first, briefly, I will be talking about what is the DDX helicase enzyme or RNA helicase enzyme, and why this is important for drug discovery. And then I will be talking a little bit about the general strategy and steps of the Transcreener assay, and then how we develop this assay, RNA helicase enzyme assay, and then what is the validation and efficiency of this Transcreener ADP assay. And then I will be summarized, and as I said, after that, I will be happy to answer your questions. So what is the DDX enzyme or RNA helicase enzyme or dead box helicases? So dead box helicase, which is named as the aspartate, glutamate, these four dead box sequence, it's a part of the superfamily 2 of the helicase family. And so far, so far 95 helicases out there, so far known, and 64 of them are RNA helicases and 31 are DNA helicases. And so far 36 dead box helicase was discovered and is going on. And then 14 are DEAH box enzymes. Also, I will be talking about the RIG1 and MDA5 enzyme, which is part of the DDX, as you see here, DDX58 and DDX116. So as you see here, these dead box helicases has shared a common motif, common structural feature. And you see the left and the right, there's a 2 N terminus and the C terminus, which is they act as they participate the protein-protein interaction, and the middle there are 2 domain, recollect domain 1 and domain 2. So, and you see the ATP binding and RNA unwinding and ATP hydrolysis and annealing all are done by these 2 domain. So domain Q1 and 2 and 4 are part of they participate in ATP hydrolysis. I'm sorry, let me. So yeah, so Q1 and then Q1, 2, and then the 6 part of ATP binding and hydrolysis, and then 3, as you see here, 3, 5, and then 6 again they participate RNA unwinding, and then motif 1A, B, C, 1C, and then 4A, and the 5, they participate RNA binding and annealing, and then 3 and 5 and 3, 5, they participate in the RNA unwinding, and then 3 and then 6, they participate in the coordination between ATP and RNA binding. So in summary is all these 36 RNA helicase and a DEAH box enzyme shares a common motif, which has 2 N terminus and C terminus that participates protein-protein interaction, and that they have 2 domains, and that different motifs participate in ATP binding, unwinding, and annealing, and the main catalytic enzymatic activity. So, what is the general mechanistic structure, mechanistic feature of these DDX helicase enzyme? So, as I showed you before, it has ATP binding site and it has 2 domains. So this is N terminus, for example, this is free enzyme N terminus and C terminus, and this 2 domain. When ATP binds, after ATP binds here, it changes. It changes to the open formation to closed formation, and which this closed formation enables extended RNA binding surface, and this extended RNA binding surface then binds the RNA substrate by their phosphate group. So, and then after binding, this substrate makes an arc feature, arc structure, and which this changing the structure of this RNA, only one strand can bind to the enzyme. Another strand is the other side, which cannot bind. So after that, the duplex RNA, this enzyme displaces one strand and leaves to the just only single strand bind to the enzyme. And after that, ATP get hydrolyzed to ADP, and at that point breaks the closed formation and to close formation to open formation, and it get back to this original formation. So main is very close. Mainly 3 steps. ATP binds, makes a closed formation, then RNA binds to enzyme, RNA displacement happens, and then it gets hydrolyzed. So that's the general mechanistic feature. So we are not going too deep into the mechanistic feature, but this is just to show you how all these dead box helicase enzyme is behaved. One important feature is these RNA helicases differ from the other helicases. So other helicases in general, they unwind the substrate from 1 by 1, 3 to 5 manner, 3' to 5 manner, but this RNA helicase unwinds this RNA helicase in single displacement. So this activity of enzyme we can measure by ATPase or unwinding assay. So 2 different ways we can measure. Here I am showing only ATPase, so using Transcreener ADP assay. So now the question is why DDX is important, why this RNA helicase is important for drug development, drug discovery, why scientists are getting interested for these RNA helicases. So as you see here, there are lots of papers. I am just showing, for example, here 2-3 papers, but last 2 decades, after discovering like 3 decades ago, especially recently, it's getting huge interest because this DDX helicase enzyme participates several multifunctional and these are multifunctional and participate several biological processes. You can say virtually every aspect of RNA metabolism, like as you see here, pre-mRNA splicing, microRNA processing, export, editing, everything. Not only that, they act as a transcriptional co-regulators and regulators of post-translation modification, and these are very critical for modulating several cellular signaling pathway. So also this DDX helicase enzyme very harmful or very have very deleterious effect on normal cellular homeostasis. So these are very important for the tumor and the cancer. And also, not only that, as you see here, the antiviral effect. So some DDX protein promotes viral replication, and some act as a viral nucleic acid sensor and promotes antiviral host response. Also, this antiviral and cancer immune, it also participate or is responsible for programmed cell death. Is several dead box helicases have been reported to drive pyroptosis, which is they're responsible for pro-inflammatory programmed cell death pathway. So all these reasons, all these RNA metabolism and all the antiviral and the programmed cell death, these scientists are getting very interested in this RNA helicase enzyme. Now, let's get back to the development. As we know, the enzymes and what it does and how it behaves, why it's important now, I'm talking about the platform we used to develop this enzyme, which is Transcreener ADP assay. Now, Transcreener ADP assay, you can, as you see here, we have three different readouts. FP, you can measure FP, which is fluorescence polarization, FI, which is fluorescence intensity, and TR fret readouts. Now, as I showed you, the general mechanism of this RNA helicase, which is ATP and the substrate double-strand RNA. General mechanism is ATP. By this enzyme, ATP get hydrolyzed to ADP, and then RNA, double-strand RNA becomes single-strand RNA. So that's the general mechanistic mechanism for the enzyme. Now, what is the mechanism for Transcreener assay? As you see here, for the FP, so you have antibody and tracer. And when ADP produced from this reaction, ADP binds the antibody and signal gets lower. For the TR fret, we have antibody terbium and the tracer. When they are without ADP, they're closely bound and you get the fret signal. When ADP is produced from this reaction, this binds with the antibody terbium and the signal get decreased. And for the intensity, we have designed this antibody and the quencher and the tracer. So when there is no antibody, there's no signal for this quencher. The ADP produced and binds with this antibody quencher, and we get the high intensity. So that's the general mechanism for the Transcreener ADP assay. We use this platform to develop this assay. So this is, as I say, this is very robust, very sensitive, very useful, very quick assay. So as you see here, so this is 5 microliter enzyme, 5 microliter substrate mix. You have 10 microliter reaction in 384. You can change this volume for 96 well plate or the smaller well. So this is for 384 well plate, which is where we use 5 microliter helic enzyme and 5 microliter substrate, 10 microliter reaction mix. Let the reaction in 60 minutes. Add ADP detection mix, the Transcreener ADP detection mix, total 20 microliter. Let the mix for another about an hour and read the plate. So this is very quick, very easy, homogeneous assay, simple mix and read format. And this is far red fluorescence readout, which is very useful for the to minimize the compound interference. So those who know that this is because this is mix and read and homogeneous assay, this is very useful for the compound screening and to run the dose response curve. So again, this is a general feature of this whole assay. The first part is, for example, this is for DDX1 and solution as a system. You have the 380 oil plate, substrate, buffer, we all provide that, and then enzyme. reaction happens, ATP produced from the reaction, and this ADP changes the signal. You get the signal and you analyze the data. Same thing here for MDA5, similar. You have the plate, substrate, different substrate for MDA5 though, and an assay buffer, and then enzyme, ATP. You get the ADP as a product, and then you have the signal reduced or increased depending on the which readout you are using. So now the development of the whole assay. So these RNA helicase enzyme, I have developed these 5 different DDX enzymes, DDX1, 3, 5, 17, and 41, and also along with that RIG1 and MD5. So most of the protein are expressed and expressed in insect cell, and most of the full length, but we also have truncated depending on the disorder domain. We, for example, here for the 3, 5, 17, and MD5, we deleted some disorder region, but we kept, as you see, the 2 domain to make it active definitely. So assay condition in general, we use subsaturating ATP and saturating the substrate to make sure the detection ATP competitive inhibitors, which bias against the RNA binders. And most of the cases, all cases except 41, ATP hydrolyze. Hydrolysis were strictly dependent on RNA. Also DDX41 also dependent on the substrate. We have this study. There are lots of stimulation for the substrate, but is less dependent compared to the other DDXs. So now assay optimization to get the final assay. So to optimize the assay, I did optimize different parameters, but like buffers, pH, different salt, temperature, everything. I'm not showing all these processes, but here I'm just showing the important feature, for example, the substrate optimization, how we get the best or better substrate. So here, for example, I'm showing you here, as you see for DDX5, I have tried different substrates here. And clearly you see enzyme titration, you see for the yeast RNA, you get the best active enzyme. So the goal is to get the best substrate to get the best enzymatic activity. So here, all the substrate gave the activity, but yeast RNA for the DDX5 provide the best activity. Same thing, MD5 for different substrate. You see here, but high molecular weight poly-IC give the best activity. And the RIG1 substrate, also try all 4, and 5' triple phosphate dsRNA give the best activity. So after buffer and everything, the substrate optimization, and also we have done the ATP optimization or KM to determine the KM of the ATP. So for example, here for DDX17, you see we have run the experiment to determine the KM for the ATP. Though this is one I would like to clarify here, this is not the full curve, and I run only 1,000 micromolar, 1 millimolar, because our assay is compatible until 1 millimolar. We couldn't go further. So the KM value is higher, literature value is really high. So for the DDX enzyme, and also for the ATP determination for, ATP KM determination for the MD5, you see it's almost full curve, and we get the ATP. So we use, in case of really higher KM for ATP, like this one, we use 100 micromolar ATP, or in case lower, below 100, we use the KM value for ATP. We have, as I mentioned again, we have optimized pH, salt, metals, temperature, time, and everything. I'm not going to show, I just showed you here the substrate optimization and the ATP KM determination. So after optimizing, this is the final titration, because this is important to get the initial velocity concentration, how much enzyme I need to use to perform this screening or the dose response curve to get the initial velocity concentration. So here you see that different enzyme titration using the optimized condition. So this is, as you see, high enzyme concentration, big signal change, MP. So you see this is a huge shift in MP, so which gives a very robust signal, very robust assay. So assay window is really high. So that means our Z prime definitely for the assay is very high. I will show you later. But here you see this is 200 millip shift. For rig 1, this is more than 150 shift, and this one also more than 150, almost 200 shift. And also we controlled no RNA. So as I showed, this is RNA-dependent, substrate-dependent activity of the activity. So you see no RNA, there is no activity here. And so after this final titration, you get the EC80. There's the nanomolar we use for the later part of the enzyme, for the screening. So this is the final titration. We get the EC80, we get the good signal. Now we use the standard curve to convert this data to product formation. So this is the ADP formation. Now this is the straight line. So this is the after the conversion using the standard curve, we get the ADP formation versus this enzyme. Now here I am showing you not only the FP, but also FP, TRFET, and FI because this is important. As I showed, the Transcreener assay is compatible and you can useful for all these three readouts, FP, TRFET, and FI assay. And you can clearly see here the product formation, and for all these three readouts, these are very, very comparable and very consistent among three readouts. DDX1, rig 1, all are very, very compatible. So you can use any readouts, depending on whatever you prefer, whatever you like to use. So we have optimized condition, we have enzyme amount, we have good assay window. Now we're getting data to run the screen and dose response curve. So here, for an example, I'm showing you the screening for the library. We use usually here I'm showing TROCRIS2 library, which has 1280 compounds. And as you see here, so this line indicates above this line, these are the hits, probable hits, and we have the controls, different controls. So from here, and this data shows the interference. We ran the separate assay to see the signal, is it for interference or is it for real hit? And you see here, this one, so these two, so these compounds is probable interfere. So this is not the hit one. So after getting the interference, and then I ran the dose response curve. Here I will show you these two compounds I ran for the dose response curve, these two compounds. I didn't do all this, but for example, I will show you these two. And here you see this from the screening, number of hit compounds, number of interference, and the Z prime for this assay. As I mentioned before, our assay window is really high, and the Z prime over 0.9. As those of you who knows that over 0.7 considered as a robust assay, we get most of the time we get 0.9 over 0.8 or something. So it's very robust assay. So now we get the screening, and then let me show some dose response curve here. So as we mentioned, the two compounds remain as a control compound. You see the dose response curve. And not only that, I have run also in experiment to see if these compounds, is it aggregator or real compounds. So you see here, I ran this dose response curve. This is normal condition with the enzyme EC80 concentration. This was 10 times higher EC80 concentration. And then high triton or detergent. Here we use 0.01% detergent. Here we use 0.1%, which is 10 times higher. And this is the summary you can see here, the IC50 of these compounds. that have the IC50 chains. So our assay is not only good for screening, but also you can run this dose response curve and also this triazine experiment to determine the compounds is real heat or the aggregators. So it's validated our assay. You can use this assay to validate your compounds. So that's all the data I want to show. Here's the summary. So this Transcreener ADP assay is, as I said, direct, very homogeneous, very easy to use, user-friendly. You can detect RNA-dependent ADP formation by different substrate. Different RNA helicases, different DDXs, RIG1 and the MD5 using FP, FI, TRFRET readouts. All these three readouts formats are comparable, very consistent. As I showed, the rate are same. You can use any one of these three readouts format. The assay provides excellent data quality, providing that the Z prime is over more than 0.8, most of the cases 0.9, good signal, good signal window, so which makes this assay a very robust, high throughput assay. We did the pilot screening, as I showed you. You get the heat from this screening, and we validated the heat compounds by doing the dose response curve. And you can get the you can get the IC50 value from the dose response curve. So in general, the summary is using this assay platform, using this RNA assay, you can get very good, you can get the platform that will help you to discover the biological relevant inhibitors of several DDXs and RIG1 and MDA5, and related, not only that, related ADP producing enzymes. With that, I am concluding my talk. Now I am happy to get your questions.
Hi, Mabat. Thank you for that presentation. Now we'll get into some questions here from some of the attendees. We'll just start with the first one here. Are there any advantages of using one assay format or over the others, FP or FI or TRFRET?
Thank you. Thank you for the questions. As I showed you, the data, our data, FP, FI, and TRFRET is very consistent. You can use any format you want, depending on what instrument you have in your lab, depending on what instrument you are comfortable with. So there is not advantage, but most of the development here, we prefer to do the FP because we think that FP is most simplistic form, simplistic and easy to use. But again, I'm not going to say this is the best one because all are very consistent and comparable. So we can provide any, we have all these materials and reagent to run any assay, whatever you prefer. So yeah, all are, we can say all are very equally compatible, but I would say maybe FP would be a little bit easier compared to others. Thank you.
Great. Another question is, you use the assay in an endpoint format. What makes it different from the universal ADP-GLOW assay?
Oh yeah, sure. So this is, yes, this is a good point. This is endpoint assay, but you can use also the current, without endpoints. So you can also use at a timely manner. So these endpoint we use usually high concentration of ATP. If you want, you can use the, what can I say? Not endpoint, so with time dependent manner. So ADP-GLOW, is, I would say, it's a similar approach. ADP-GLOW also you can use. It's not different, but different in a sense they use a definitely different reagent. But again, you can use our method. You can use as a stop method. Endpoint, or also you can use continuous method. So both are equally possible and equally useful.
Okay. Did you, as part of your studies, did you try to use the assay in kinetic format or measure the linearity of the reactions in a time frame you were proposing?
Yes, we do. I mean, I did. I mean, definitely. It's not I'm just showing here the only endpoint assay. I have the data. It's kinetic 1 hour, 2 hour, 3 hours. We have you can measure in 15 minutes or 20 minutes. So definitely this is possible. Yes, I have the data. We do, and I did that. So great. Yeah.
Another question we have here on the topic of potential assay interferers, given that the activity is triggered by RNA substrate DDX interaction, how are false positive hits, which disrupt the protein oligo interaction, identified? Did you include a counter screen for RNA binding?
For that RNA, we didn't. Honestly, we didn't do that, but it's possible. I can tell there's possible that you can run that definitely. Our RNA substrate is not in general. It's not inhibiting. It's not interfering our assay. We have the data. When we do the assay development, I try this, that how interfere the different substrate with the assay. It's not interfering. So if you want to check the substrate interference or counter interference, definitely is possible. But so far, these East RNA or the other substrate, or these are not interfering with the assay.
Along those lines, could you elaborate on how you found the false positives in your assay for the screening?
So here, as I showed, I mean, so these are the data, this is validated data. So you can, for example, if it's false positive, it's not good hit. So this simple experiment, I mean, you can easily determine, primarily, you can easily determine that this tries an experiment. Is it a false positive? Is it aggregator? Is it non-stoichiometric inhibitors or stoichiometric inhibitors? So as I said, that's why, for example, I showed you here this with high DDX, high enzyme concentration, dose response curve, and high triton. So you can, by doing that, you can measure primarily measure that is it, as I said, is it aggregator, is it false positive or not. Also, we're able to do resident time and other assay to confirm is it false positive or not. So, and primarily, as I showed, this is the interference assay. From here, if you compare this interference, definitely. So we exclude this interference. Also, another one we found among these RNA, we found one universal, the promiscuous compounds. These compounds probably the promiscuous compounds we found because this was showing this SCH202676. It seems to be promiscuous, and the hill slope is very high for that compound. So by doing all these, you can get an idea that this is false positive or aggregator or promiscuous or not.
Going back then to your interference screen, could you determine or describe, I guess, the difference between these two graphs?
Yeah, sure. So this one we ran in EC8 with enzyme and the substrate. So first we have the compounds library or the plates. We add the enzyme, EC8 amount of enzyme. We incubate with the compounds to make a enzyme and inhibitors complex. And then after half an hour, we add the substrate to run the assay at usually 1 hour, and let's measure the ADP, and then we get the signal. And this one, so as I mentioned before, maybe you can remember, our assay is initial velocity, so we maintain the initial velocity amount. So that means we prefer to get less than 10% conversion. So here, usually we take only 10% conversion. That means 10% product, no enzyme, and then the compounds. If compounds, to determine if the compounds interfere with the assay compound with our transcreener assay reagent or the substrate or ADP or anything. So this definitely differentiate what is interfere and what is not. I hope you get your answer.
Excellent, excellent. Well, that's all the time we have today. With any other questions, please contact us by email at info@bellbricklabs.com.